Okay, I've got 2:01 Eastern, 3:01 Atlantic. So let's get kicked off. Welcome to the Sona Nanotech webinar. I'm David Regan, CEO of Sona Nanotech, where we're developing a nanomedical therapy for cancer. And I'm delighted to have with me today not only my colleague Len Pagliaro, PhD, who is our Chief Scientific Officer, but also Drs. Carman, Giacomantonio, and Barry Kennedy from the Giacomantonio Research Group. When worlds collide, innovation can happen. And that's exactly happened to us when Sona's nanotechnology world collided with Dr. Giacomantonio's research group's immunology world, leading to a number of revelations amongst us and opening up some possibilities that could change the course of cancer research. Today, we're excited to be able to share with you some insights from Drs. Giacomantonio and Kennedy and provide a high-level view on the potentially groundbreaking study that we will undertake together and what those results could mean. Let's go down a page. So, of course, we are a preclinical company, and we're going to make a number of forward-looking statements today, so we'll seek safe harbor from that. By way of background, Dr. Carman Giacomantonio is a surgical oncologist at the QE2 Health Center, professor of surgery at Dalhousie University, and senior scientist at Beatrice Hunter Cancer Research Institute, a great entity, by the way. Dr. Kennedy is principal research scientist with the Giacomantonio Research Group and was formerly a research scientist with a clinical stage biopharma specializing in cancer vaccines, which is apropos given some of the conversation you'll hear today. Before we hand it over to Drs. Giacomantonio and Kennedy, I'm going to let Len, our Chief Scientific Officer, take us through a very quick overview of Sona's burgeoning and developing cancer therapy. Len.
Thanks, David. Basically, what we're developing is a two-component medical device system for applying therapeutic heat to tumors, to solid tumors. Therapeutic heat does a number of good things for tumors, and you'll hear more about that. But basically, the way it works is that we inject nanoparticles, and the image on the left shows a red blood cell. That's that big red orb in the middle of the slide. And the little tiny specks in it, you can see only in an electron microscope. Those are nanorods. They're precision gold nanorods. They absorb infrared light, not radiation, infrared light, and they emit it as heat very efficiently. Our initial therapeutic target is colon cancer, colorectal cancer, specifically rectal cancer, a large unmet need, easily approachable for administration of the infrared light using a scope, a flexible sigmoidoscope or a colonoscope to get the infrared light to the target. The nanorods are injected systemically. They circulate through the body. They concentrate in tumors because of the leakiness of tumor vasculature. And what we wind up with is shown on the right, where we can illuminate the region with a tumor or tumors with infrared light and generate that therapeutic heat that emanates from within the tumor.
So we're going through this obviously very quickly. Len and I are always happy to chat with investors and talk you through this, and I can refer you to a few other full presentations we've done on the concept, but we want to skip right along to the main event here. And so I'd like to hand it over to Dr. Giacomantonio.
Well, thanks, David. So this is truly an exciting opportunity, and as David said, a collision of two worlds. So just by a bit of background, the immune world I work in, most of the world is aware of what cancer immunotherapy is now, and the kinds of therapy that most people are aware of are the systemically delivered checkpoint inhibitors. These are monoclonal antibodies that are targeting cells that have already engaged the cancer. So in the normal course of things, a cancer is seen by your immune system, but because the cancer also has either a weak message, the way the cancer is seen is called an antigen, and the antigen is weak, and so the response isn't robust, or the cancer also gives signal that it's self, and so your immune system, our evolution is that the immune system won't attack self, and it stops attacking self by turning on these checkpoints, and the checkpoints are doing their job by preventing your immune system from killing what would be considered self, even if that self happens to be a cancer. So what the most exciting checkpoint inhibitor, the pembrolizumab, that was the Nobel Prize got awarded for, was essentially something to block that message, and that creates effective immunity. But it doesn't. That immunity, what we're taking advantage of there is the knowledge that the immune engagement has already happened. So for a checkpoint inhibitor to work, for those monoclonal antibodies to work, you need to have an engaged system. So the failing there is if the system isn't seeing the tumor, then nothing's going to happen. The other thing on CAR T cells is very exciting in the blood cancers, not as effective in the solid organ cancers like colon cancer. There's a number of reasons why, and the biology of CAR T is too complicated perhaps just to go into today. But the idea is that as I said, when a cancer is seen by the immune system, cells invade that cancer. And what a CAR T cell is, some of those cells are taken out and the receptors are expanded, and then they're put back into the system. But the process of doing it is onerous. And again, the effect in solid organs or solid cancers hasn't quite been as exciting as the blood-borne things. And then cytokine treatment, which is what I do. So this is where our worlds collide. The tumor microenvironment, the cancer, when a cancer grows into a lump, a tumor, it creates a microenvironment. And that microenvironment, you consider, it's like, cancer is like an embryo. It's trying to grow a new thing, a new entity. And within that microenvironment, there are built-in checks and balances to prevent the immune system from penetrating it. And so the simple solution, so what I started doing 11 years ago was to take a cytokine. A cytokine is a hormone, if you will, produced by a cell that makes a cell next to it do something. And one of the most important cytokines in an immune cascade, an immune series of events, is interleukin-2. And so I take interleukin-2, and initially we used to give it intravenously to patients. And the problem with that is that it creates such a storm of immunity that many patients got too sick to tolerate the treatment. So what I recognized was I can inject that directly into the tumor and get the massive effect of a large dose of interleukin-2 on the immune system in the microenvironment of the tumor and save the systemic toxicity. And that's exactly what we've achieved. What I'm doing is exactly what the checkpoint inhibitors do, except I'm driving the system versus the checkpoint inhibitor, which is unbridling the system. The problem is that the interleukin-2 alone also requires an immune engagement to have occurred. The immune system has to be recognized, and those cells have to be poised and ready to attack. And then in the context of interleukin-2, the cells will be very aggressive and overwhelm any inhibition, i.e. checkpoints and other things. And so that's the kind of, but we've been limited. So some of the things we try to do is try to generate new immunity. And that's where this technology holds all the promise. What we've recognized over the years is from time to time, when we give interleukin-2 into a tumor and we use some other immune stimulant, in my case, sometimes we use a tuberculosis vaccine. So if I vaccinate the tumor, I create an immune response. And what happens is sometimes that process generates new antigen. The tumor becomes all of a sudden newly visible to the immune system. So 3 out of 10 times I do that, we see this response we call abscopal, meaning I treat a single tumor or a single area of a tumor, and all the tumor or all of the metastatic cells from the tumor die because you've generated new immunity. And that's what the nanoparticle can do. So when we say it boosts the immune system, it does more than boost it. It actually exposes new tumor antigen, and that is literally the holy grail of immune therapy. And so that if I can stimulate a tumor to expose new antigen, and the way that happens is through the trauma, the heat generated by activating or exciting that nanoparticle. When the tumor is heated, a number of things happen. Some cells die because they're fragile and they go into a death cycle. The trauma alone disassociates the cells, And a lot of these, when the immune microenvironment is perturbed, a lot of the balances and checks that are preventing additional immunity or a number of other things from happening are upset. And so it's the notion of disturbing or perturbing the tumor microenvironment that makes this highly valuable. And we see this in other areas too. From time to time, when a patient has a cancer and we give them immunotherapy and it's not working, but then we irradiate the cancer to try to kill it, that radiation causes the cancer to die, and the dying cells are re-engaged to the immune system, sometimes showing these new antigens. And this is called an abscopal response. And the problem, of course, with radiation is that it's highly toxic, and it's a very difficult thing to deliver selectively to a particular area. Doesn't mean it doesn't happen, but it's a far more onerous situation. In this case, if we were to deliver the nanoparticles systemically and they accumulate in a tumor, then we can selectively activate where those nanoparticles are and spare the toxicity to the surrounding tissue. And if we do that and we take advantage of stimulating and overstimulating the immune system in that microenvironment, there's a very strong possibility we'll generate new immunity. And that's the exciting part of this technology.
So you can see this then taking the place of radiation in terms of providing that, perturbing the tumor environment?
Well, in many tumors, yes. So the answer, if you looked at a colon cancer or an early colon cancer where you wouldn't irradiate, for instance, a rectal cancer, you can radiate, but if you had a colon cancer outside the rectum, to deliver radiation to that tumor is too toxic because all of the surrounding bowel that's in, just sitting there passively would be also irradiated, and that toxicity is too much. And so having the ability to flood the tumor with the nanoparticles and then selectively excite that piece of anatomy without the collateral damage is really, it just opens up all kinds of opportunities. The same with gastric cancer, the same with pancreatic cancer. You can, and these are the big challenging tumors. Yeah, you definitely can. You could envision it. Anything you can. Prostate cancer, ideal because it's accessible to external excitation in a very controlled way. Esophageal cancer. These are the bad cancers that we don't have a lot of other things for. And typically, for instance, a colon cancer wouldn't be. It's sensitive to chemotherapy, but only 30 to 40% of cancers respond to chemotherapy. The ordinary garden variety colon cancer isn't typically responsive to immunotherapy. But if you were to generate potential tumor antigen using this technology, almost for sure you would see an increased response rate. And so that would be amazing, yeah.
Right. So when our worlds collided, we talked about the idea that we were going down the path of targeted hyperthermia therapy as a monotherapy on its own with the goal of shrinking tumors. You very much opened our eyes and looked at the possibilities. And you've described it in the past as combining the two could provide a 1-2 punch. And I'm going to flip to our next page because I think we're basically in that territory anyway now. Yeah. But can you speak a little bit to what why that's important to provide that combination?
Yeah. So that's essentially combining the two means the logistics of heating the tumor, exciting the tumor, causing tumor destruction, necrosis, which is cell death, exposes a myriad of antigens, tumor markers that the immune system can now see that couldn't see before. And that followed by, or immediately followed by, an immune stimulant is the hypothesis that we're fairly confident we'll see results with. Is that so? This you're weakening the tumor, but you're not only weakening, you're destroying the tumor. That's what the local nanoparticle will do. So it's very effective on its own. How complete the tumor kill is generally the challenge. And so that immune 1-2 punch, that second effect is critical. And then again, ideally, when the immune system sees a cell or a cancer antigen, it generates immunity in the lymph nodes, not right in the tumor. And so those lymphocytes, those cells that come out to kill that tumor, have to actually circulate through the body to get back to the place, the antigens that it sees and recognizes. And so other cells that are distant would also be seen by that tumor. And that's that, again, by that immune system. And that, again, is that abscopal effect that we think this can generate.
So what would that mean then if it's traced back to the lymph nodes and recognized in different parts of the body?
Well, so, so if it's, say, a colon cancer, but it could be a skin cancer or a breast cancer, when a tumor is dying and you stimulate your immune system to clean up or to focus on the new antigens, the antigens that are presented from the tumor shown to the immune system are actually shown in the regional lymph node. So the lymph node closest to the tumor is going to be where those T cells, those cytotoxic cells, are actually finally activated. Some happens in the tumor, but the vast majority happens in the lymph node. And then the cell has to leave the lymph node. When it leaves the lymph node, it has to go through the venous circulation and up to the heart and all that stuff. It has to get pumped, circulated to the body. So those cells are circulating literally in every part of the body. So if there is a cell, a tumor cell that's gone metastatic, it's gone off to another part of the body, and it has the same antigens, it has the same signals as the primary tumor, well, that cell will be attacked by that immune cell. And so it's effectively creating a systemic response generated through a local engagement.
So even if it was just one tumor in one area of the body that was treated, it could have benefits for other tumors of other types of cancer throughout?
Exactly. The same cancer, but in other areas. So, and as we know, a colon cancer spreads to liver, to lungs. And so that's all part of the systemic circulation. And so in a true systemic response, the primary cancer should respond, but so should the metastasis, all of those other cells, because they're provided they're the same cells. Now, there's little caveats around that, we can, those that don't respond to this, if the primary tumor responds and only some of the metastases respond, you can reproduce this process by then targeting the metastasis, which the assumption is that those metastases, those distant cancers, are subtly different. So they're a subgroup of that cancer. And so we know that there's a term called immune editing that people in the business would be saying, well, that's why this single treatment didn't work. But in generating novel antigen, you should be able to overcome that barrier. And so this 1-2 punch is critical to the whole thing happening.
Okay, and look, I've received a question here. There's a bit of a webinar chat box, folks. There's a button, I think, at the top of your screen. If you have questions, there's a question here as to whether this would enable targeted hyperthermia or immune therapies to be applied to later stages of cancers that are entered into later stages. Does that have any impact?
Oh, absolutely. So this is, and that's, again, a cancer that's been able to progress to a later stage has evaded the immune system. May not have ever been terribly immunogenic. What you need to do is engage, what we need to do is engage the immune system somehow. Cell death and necrosis, like that radiation story, is the perfect way to do it. So it shouldn't matter what stage the cancer is at. Historically in cancer treatment, we've always looked at new therapies and taken the worst situations as our entry-level cancer, which is kind of a mistake if you think about it, because the more cancer burden, the sicker the person, the less likely for an effective immune response, which is called anergy. But certainly a cancer that has a limited amount of metastasis in a healthy person, otherwise healthy person, you would think you can engage that system anywhere. So our hope would be to try this in the earlier stage cancers. And so there's little downside to it. The toxicity, the way this is delivered, the way the nanoparticle is delivered and activated, it's very minimal toxicity relative to so many of the other things we do right now.
Certainly, and that's certainly what Sona brings to bear in terms of the particular gold nanorods we create and the patent on on their biocompatibility and that they don't use toxins, therefore lending themselves to in vivo application. And of course, we've had a great deal of analysis done by the NCL we've talked about in the past. Another question here, if we're talking about a two-step treatment with targeted hyperthermia first and then immunotherapy second to finish the job, then isn't immunotherapy success dependent on targeted hyperthermia therapy success first? That is, we need to wait several years before this therapy can be successfully introduced before it can be used.
Well, I mean, yeah, I understand the question, but I think that I think that we've got fairly compelling evidence that this hyperthermia is effective at causing local tissue destruction. And that's what some of the modeling that we're going to do, the animal modeling will do, is to not only prove that it has that, but understand the details of what that destruction looks like. It's we don't have to. I think we already have immunotherapy introduced into the healthcare system in a number of ways: systemic, direct intralesional injection, and so I think these things will naturally dovetail. Once we've got the hyperthermia, once we've shown that we can safely induce hyperthermia in a lesion in a bowel, or in a lesion in the skin, or a lesion in a breast, or what have you. The rest of the immunotherapy isn't. I think that can be done coincidentally as this we call an adjuvant study.
Indeed, which is kind of a segue into a little discussion perhaps around what these experiments in the study that we're going to undertake look like, and then that ties into a question we've received here about what, if any, have been the roadblocks in your research to date.
Well, the biggest from the immunotherapy, the biggest roadblocks are, first of all, these are animal models, and so we have to be very careful that we don't equate one clearly with the human situation. But the biological principles are sound, and so so generating tumors that mimic, to the best of our ability, the kind of cancer that we're interested in treating. So many of the tumors we introduce the tumor to the animal by taking cancer and then implanting it in an animal that we know will tolerate it. So these are genetically selected animals, mice, and then treating that tumor. It's a bit of an artificial situation, but we can watch the response from the nanoparticle, for instance, by measuring and analyzing the tumor before and after treatment, and in conjunction with the rest of the animal. And then we can look at the immunotherapy. But the immunotherapy delivering it, when it comes to these things, one of our challenges is understanding what's the appropriate dose relative to the response. These aren't easily translatable to the clinical situation. That's why the clinical trial has the phase 1 of a clinical trial is that dosing challenge. It's the same in the animals. Otherwise, the only other challenges are logistics. It's getting the research done in a timely fashion. These are very big experiments that require repeat, and so time is our other challenge, really.
That's certainly it, and time relates to the data required to support this and show what sort of a half-life there is in the materials that are injected, and where there's concentrations of it. This is all information that regulators would want to know. I have a question here about the sort of regulatory pathways, which isn't necessarily germane to this, but whether something like this can be fast-tracked, it's unclear as to that, whether that's the case. But certainly combining it with an approved immunotherapy is going to be helpful, because it could presumably ride on past clinical trials on the immunotherapy.
100%. Yeah, I think that's the reality. This is really functioning as a device, and so I think that's a more straightforward pathway in terms of introducing it into clinical trials, and all of the things it would be combined with are already approved. So this is in terms of logistics about getting this into the clinic, I don't think are nearly as challenging, mostly because it's a very controllable device.
Right, right. Len, did you want to comment anything on that in terms of the, the therapy as a device, being classified as a device?
Sure, I think that is an important aspect of what we're doing. Typically, I don't think most people view an injectable material as a medical device, but there are precedents for it. For example, some of the contrast agents used for MRI are injectable medical devices, and there are some other injectable metallic nanoparticle medical devices that are in the clinic in the US. They're not yet to market. They're getting closer, but they're in the clinic in the US. And so this is a relatively new field, but I think it's going to become a very big field and an important field. And as a medical device, if you show a good safety profile, which we have an excellent safety profile so far based on preclinical work, then you have a significantly less onerous time and cost in terms of the clinical work you need to do to get to market.
Yeah, that is key.
And part of the research that we're planning to do is to look at whole body distribution and tumor concentration. So we should be able to give some fairly high level detail about what the collateral exposure is, but so recognizing that
where the nanoparticles would aggregate and remain, and how quickly they'd clear.
Yeah, exactly. Yeah, so that's some of the information we're going to look at. And the other thing is recognizing that this particle is essentially inert unless it's excited, and the exciting agent, the near red technology that we're going to use has to be held juxtaposed to the area of concentration.
Got another question here on whether this could potentially become a cancer vaccine.
Well, that's a great question. It potentially enables the development of a cancer vaccine. Cancer vaccine, the true vaccines that are being developed now are personalized, and so the idea is that, again, vaccines have failed in cancer therapy horribly. There's a famous surgeon scientist, Don Morton, has spent his entire life and fortune trying to create a vaccine against melanoma. And the reason it failed was because the melanoma antigen wasn't significant, wasn't robust enough. The cancer wasn't strong enough to generate an immune response that overwhelmed all of the checkpoints and generated memory so the disease didn't come back. This nanoparticle will expose tumor antigen that the immune system hasn't seen, that your body hasn't seen. And so when you do that, you potentially create a personalized vaccine. And this is future thinking and future planning, but in the future, we'll be taking the cells that are generated, the immune cells that are generated from this process, and expanding them, kind of a reverse CAR-T, making this a doable thing, because we're taking your own T cells, expanding them and reintroducing them into the same person, so they won't cause rejection. We won't have to immunosuppress that patient to make it work. So yeah, this is
a personalized vaccine is what that sounds like.
Exactly right. That's exactly what that is.
I have a question here also on, is anyone else trying to do similar cancer treatment with gold nanoparticles? Will you be the only ones testing this technology? And Len, you can perhaps speak to that for a second, but also that opens up the question of, to your knowledge, is anyone else doing this type of research in terms of a combination of using injectable nanoparticles, combining them with immunotherapy? So first to Len, perhaps, and then to Carman.
Sure. Well, we do know at least one company in the US that has a nanoparticle, a metallic nanoparticle for injection combined with infrared light, called Nanospectra Biosciences. They are in the clinic in the US, not yet to market. We believe we have multiple advantages over their technology. Their technology is 20 plus years old. We have nanorods instead of these very large spherical nanoparticles. We're using what we believe is going to be a much more efficient coupling of the light device to the nanoparticles, and a better ability to guide the light device to where it needs to get in the lumen of the gut, for example. And so there are other examples of that out there. In FDA terminology, they're not yet predicates because they have to be to market to be a predicate in FDA terminology, but they are in the pipeline to get to market eventually.
Yeah, and in the clinical arena, there's precious little actually happening that clinicians are engaged with or aware of in this with this kind of technology. So there's plenty of space for this.
Excellent. I've got another question here asking as to how long the study is expected to take.
That's what I said. So time is the challenge. It can be our barrier. So we hope we're going to have early data probably in December, and we're hoping by February we have publishable data on 2 of the 3 arms of our initial plan. So yeah, we things are well underway in terms of the animal ethics and the lab preparation, and we're hoping to start to have mice in-house mid-October and then go from there.
Right. Now, I'll just move forward to the next page. I think this takes us to it, and that raises the question. You mentioned on some of the implications and what happens. You mentioned publishing. What happens with the data that comes out of this study?
Well, again, we hope it's very positive, and we can use it to really generate the next series of research. If this does what we expect it to do, that is to generate stronger novel immunity, then that's going to be published right away, because we want people to know that there's a better way to generate immunity. And then the next thing is it'll lead to, we hope to some powerful peer-reviewed grants that will let us take this to the next level, which is to start to look at vaccine development.
Right. Could you name sort of what sort of journals would this go into, or are there conferences that you'd aim?
Oh yeah, so I mean, the journals at the level of things like Frontiers in Immunology would be one of the cancers and cancer research. Those are the kind of scientific clinical journals that overlap, that are very credible. It's once we get to the point where we're looking at true cancer vaccine, we would take it higher, and we'd start to go to Journal of Immunology and these more elite publications. It just depends on most modeling. Most modeling is exciting, and proof of concept is exciting. The higher the journal publication, the more complex the science needs to be. I mean, everybody wants to publish in Nature Medicine, but to conduct those experiments is hugely expensive. So to get from here to there, we need compelling data that would justify the kind of CIHR grants and investment that we can anticipate with some confidence that we would have the data to go to the next level publication. Which
presumably would be a more complex study or set of studies to warrant the more publication.
Yeah, exactly. So that's where that gets into the molecular science of it, and of course that's the level we have to get this to. But we proof of this, the data that we'll generate come December to February will be. Should be the compelling proof of concept that we're right. We can make it happen, and if it make it happen in the big picture, then you can dissect those parts and really start to get into the science of it.
Right, and now perhaps more difficult question here, but level of confidence on achieving or testing these hypotheses and what these outcomes could be.
Oh, I'm more than confident. As I said, anything you can do to expose and present new antigen, we can exploit with immunity. There's no question this is going to have an effect in the animals. It's the magnitude of the effect, and we don't know where the pitfalls are going to be, honestly. So TBD, but I'm extremely confident that the hypothesis is sound. Antigen, the grail of immune therapy is antigen. Strong antigen, strong response, weak antigen, weak response. In the current immune therapy environment, what we're focused on, and I have concerns about, is impeding checkpoints. So in a checkpoint inhibitor works. And if you use a single inhibitor, the toxicity is not so bad. About 20% of people have severe autoimmune toxicity. So checkpoints are there for a reason. We don't want to block them and perturb them. We want to generate novel antigen your immune system doesn't see normally, and that's counter antigen. There is our bodies learned to adapt and respect itself. Autoimmunity is a disease lots of people have, and that's when that all breaks down. The more we block a checkpoint, the more we are likely to see that autoimmunity generated. This is the better solution. Just generate better antigens and don't have to unbridle the system.
Certainly works better with the system in that case. More elegant solution, it sounds like.
Absolutely, yeah. This is the solution to, block. There are some cancers we give two checkpoint inhibitors to, and we see 50% toxicity. That means we need a better solution. This is definitely a better solution.
Excellent. When we started with targeted hyperthermia, Len, I think the concept was purely just on shrinking the tumors, and we hadn't gone to that next level of creating antigens. The idea that antigens that would be created could be leveraged and used. Is that fair to say, Len?
Well, we didn't have any evidence for that. We had very good evidence for shrinkage of tumors and increased survival in animals in the laboratory. We certainly had the expectation and the hope that we may be able to generate a more global immune response, but we've never had the ability to generate data for that before. And so that's one of the things that's so powerful about this collaboration.
Yeah, that's it. Barry, any comments as we get close to closing out here? I know we haven't let you say a word edgewise here.
No. I think almost everything was covered, but I'd just like to echo a couple, I think, crucial points that Carman mentioned earlier. And what that really is with this collaboration, we're really going to delve deep into the mechanism. We're going to truly identify potential biomarkers and the mechanism of action of this immune cell infiltration into these cancers after the THT. And what we can do with that is really take that and use our knowledge and really dig into the improvement of the efficacy of this therapy. So the possibilities are endless, and I do want to just emphasize one more time that this is truly a personalized therapy, which is really what we're all looking for, to minimize side effects of any potential therapy and really aggressively target and kill patient tumors. And the goal is to use this study to make it into a bigger study as well, but to eventually translate this into the clinical setting.
That's great, and it's it ties into some of the merits of targeted hyperthermia that we're proud of and seeking to accomplish are the lack of or the absence of collateral damage, and that's exactly what you're speaking to. And when we add these two together, again, the impact is a much more selective therapy with limited to no negative ramifications and implications.
No, exactly.
Excellent. Dr. Giacomantonio, any closing words? Anything else in terms of where this goes, how it might affect other research in the future once we have results?
Well, yeah. So I mean, throughout the history of what we've done in cancer treatment, we've recognized that the final common pathway to cell death is often immune cell death. Even when people get chemotherapy, when chemotherapy causes a tumor to die through necrosis, or the tumor starts to shrink away, that debris is mopped up by the immune system. And so this immune cell death is a pathway that right now we've exploited, and we're using checkpoint inhibitors to say, well, we know the immune system works, but that checkpoint inhibitors work 40 to 60% of the time. There will be a response. That 40% of people that don't respond to the traditional checkpoint inhibitors that we're using, this may open the door for them too. This, in the context of the systemic checkpoint inhibitor and the incomplete response or the no response, using this technology to reintroduce the cancer in that context is also on the table. That's part of the future for sure. So there's a whole other world of systemic treatment that may be revisited with this technology.
Yeah, well, we're extremely excited about that. Look, we've been very fortunate to be working with you, and for your time today. We thank you, both Carman and Barry, and we would look forward to having you back again in a few months to be able to talk about some exciting results, hopefully, and shed some color on that.
I look forward to it.
All right, excellent. Thanks so much. One last question here. Expectation, results published, grants, studies starting. Well, a little bit about starting times with next studies, but you know, certainly our timeline here, made it clear, is into the winter and spring for strong results, and hopefully publishing something which would absolutely lead to you know, a next level study soon thereafter, I would think. Is that fair to say, Carman?
Exactly right. Yeah. So we'll be writing with the results that come through in December. That will be the foundation to start writing the grants. Because that's really the information we need for the granting agencies.
Right. Excellent. Well, it's exciting times, and again, very fortunate to be working with both of you, and look forward to speaking many more times.
Yeah.
Thank you.
Pleasure.
Thank you, everyone.
Have a good day.
Cheers.